All right, hi everyone. So in the prior video, we went through this intrinsic apoptosis or apoptotic signaling, started to talk about our pro-life factors, which are labeled right here. We talked about our pro-apoptosis factors, and we talked about our death helpers. In this video, we're going to talk about, okay, once we've actually formed a pore with our death helpers, which I'll form down here just in alignment with where I had it in my prior video. If we actually form a pore, what comes next? So let's say we have backs, and back in this case, we've formed a pore on our mitochondrial membrane, and we can start to actually bring proteins out of the mitochondria. Now, just to really clarify kind of what the steps are going to look like, because we talked about some of the processes last time. Before we form this pore, just to back up really quick, are two signals that are going to come in that tell us we actually need to release things from the mitochondria are caspase 8, which I talk about in the TNF and FAST signaling video, and DNA damage. So this is my extrinsic signal, and this is my intrinsic signal, right? That tell me that, okay, we need to maybe go through apoptosis. These two signals are going to directly activate the BID death helper protein. And that's going to lead to insertion of backs and back into the mitochondrial outer membrane, right? So if I have BID here, going to be interacting with these proteins and inserting them into the membrane prior to this pore formation. So this is kind of step 1, 2, and 3. And now we're able to form pores at this step, which brings us right back to where we were at the start of this video. So once we've formed our pore, what's going to happen next? So we release 3 different molecules into the cytoplasm when we form this pore, and these are stored inside the mitochondria. They are procaspase 9, cytochrome C, and SMAC Diablo. So these are the 3 molecules that are released from the mitochondria into the cytoplasm at this stage. And 2 of them in particular, procaspase 9 and cytochrome C, are going to come together along with 1 additional molecule called APAF1. Let me write all these down here. We have APF1, procaspase 9, and cytochrome C, and these come together to form something called an apoptosome. Which is the term for kind of the interaction of these 3 proteins. We're not going to go into detail about the apoptosome other than to say that the product of this apoptosome formation is going to be the production of an active caspase 9 protein. So this is our active form. So just like with procaspase 8, when we have the pro in front, this is inactive, it needs to be cleaved. Now we have our caspase 9, our active form, after the formation of this hepatosome. This leads to the executioner cascade. And apoptosis ultimately. Right? So that's kind of our interaction with the mitochondrial outer membrane and what it is going to look like for that process. Now just really briefly to zoom back in and look at some extra proteins here. We have two that are important. We have IAPs, which are going to be inhibitors of apoptosis proteins. And our IAPs are going to inhibit this conversion of procaspase 9 into caspase 9 into its active form. So that's our role of our IAPs. They're an inhibitory protein in this pathway. And then lastly, we noticed that we didn't talk about SMAC Diablo at all. SMAC Diablo's role is actually to inhibit the IAPs. So SMAC Diablo is essentially a pro-apoptotic protein. It inhibits the protein that would inhibit apoptosis. So that double negative kind of leads to this benefit for apoptosis when SMAC Diablo is present. Awesome. Okay. That's what we're going to talk about for this video. We'll go into kind of some of the ways that P53 is going to interact with this pathway in the next videos.